So this is where I am today. I am at a cafe in Dhaka, the capital of Bangladesh. It's very noisy all around me because of the construction and the traffic. And you get used to it after a while, or maybe not. But that's all right. We can still get our work done. All right. So what do I want to talk about? I want to talk about wood. Let me start with a bit of plant biology before I get into the details of the research paper I want to share with you. Auxin and cytokinin are two plant hormones that have different roles in plant growth and development. Auxin is primarily responsible for cell elongation, and it suppresses the growth of lateral bud. It's also involved in phototropism, you know how plants bend toward the light source. Auxin also helps regulate the development of roots and the formation of fruits and flower. On to cytokinin. Cytokinin is involved in cell division and growth, especially in shoot tissues. It promotes the growth of lateral buds. Remember, auxin does not. And cytokinin delays aging in leaves. The ratio of auxin to cytokinin in different parts of the plant determines what type of plant tissues will form. On to the research paper. The title of the paper is Physical, Mechanical, and Microstructural Characterization of Novel 3D Printed Tunable Lab-Grown Plant Materials Generated from Xenia elegans Cell Cultures. Dr. Ashley Beckwith is the primary author at MIT, and this research was published last year, 2022. Okay, so what is so interesting about this research? The scientists at MIT developed methodology for 3D bioprinted lab-grown wood that did not require cutting down a tree. No deforestation needed. By the way, the lab-synthesized wood, can be made in any shape or size. They took cells from the leaves of a flowering plant, the common Xenia elegans, and incubated in a medium for a couple of days. After that, they incubated the plant cells in a gel-based medium that contained nutrients and hormones. What hormones? Auxins and cytokinins. Eventually, as the new plant cells developed, the researchers observed that by changing the hormonal concentrations in the gel medium, by tweaking the concentration of auxin and cytokinin in the gel medium, they could control the physical and mechanical properties of the newly grown plant cells. Wood. Higher hormone concentrations, as it turned out, made the wood stiffer. So the composition, the structural properties changed. The research team then moved on to 3D custom designed shapes, structures, out of the cells cultured in that gel medium using a 3D bioprinting method. For 3 months, the 3D bioprinted plant material was incubated in the dark. And lo and behold, the lab synthesized wood grew at a faster rate than a regular tree. Why is this research fascinating? Well, one will be able to grow plant material, wood, without the need for land, sunlight, or trees. No land, no sunlight, no trees. And lab synthesized wood can be then produced in any part of the world. They can produce wood on location using a lot less energy, hauling of raw materials, harvesting and processing of lumber. Think about the time, land, water, fertilizers, pesticides that are used to grow whole plants. This in vitro cell culture technology using plant hormones and 3D bioprinting will be a game changer. One can produce the final product, be it a table, chair, any piece of furniture or construction material, pretty much in the final shape and size using this method. Every year, due to deforestation, we are losing 10 million hectares of forest. That's an area more than 3 times the size of the country Belgium. The trees we are losing do not grow back right away. So what do we know about trees? Trees remove carbon dioxide, CO2, from the air. Trees are carbon sinks. They sequester carbon. And we really do not want the carbon sequestered away in trees to be released back into the atmosphere due to deforestation. Polling time. I have 2 questions, yes, no, true, false answers. The secure web link will be attached to the comments or description section. Please participate. Till next time, stay inspired.